Okay, so I was wrong. We do have one more lecture. Usually the lectures aren't going to be more than four, which is 4 15-minute sessions, which totals to be an hour. But this is a long chapter, so I apologize. But anyway, we had gotten to the checkpoint questions. How does the nucleus control the cell's activities? And of course, that's because the nucleus contains DNA, and DNA contains our genetic information, genes that determine, the particular proteins that the cell produces, and those proteins determine the activities of the cell in large part. All right, what process would be affected by the lack of the enzyme RNA polymerase? All right, RNA polymerase is the enzyme involved in transcription, so I'm just going to tell you that answer. This is a lot of information, so you just kind of do the best you can with it, but you do tend to see it again on the TEAS test, so it's not only important in this class, but also for that test. But anyway, the answer to 23 is because it's involved in RNA polymerase is the enzyme that converts DNA to RNA, to messenger RNA. And then 24, during the process of transcription, a nucleotide was deleted from a messenger RNA sequence that coded for a protein. What effect would this deletion have on the amino acid sequence of the protein? Well, basically it would shift it, so the triplet codes called codons are going to code for different amino acids now. And if the amino acids are different, it's not going to be the same protein. So it could be disastrous, Now, there is a whole section on the life cycle of the cell, and you may have studied this in biology before. If you took any biology, like in high school, even if it was a long time ago, hopefully you've heard these words before. The stages of a cell's life cycle include interphase, mitosis, and cytokinesis. Cell division, which includes mitosis and cytokinesis, increases the numbers of cells. So whenever we are first created, when the sperm from our dad fertilizes the egg from our mom, that becomes a zygote. That is the very first cell that is going to divide many, many, many times into the trillions of cells that form us, okay? So those cell divisions where we increase from 1 to trillions of cells, those are called mitosis and cytokinesis. Mitosis is the part where the nucleus divides, and cytokinesis is the part where the entire cell splits in half, the cytoplasm divides. But before the cell can divide, since each new cell requires a copy of the DNA from the original cell, in fact, each new cell requires all 46 chromosomes. So our very first cell, the zygote, that divided trillions of times and formed us, that first zygote had 46 chromosomes, which contained all of our genes, and all of the cells that divided from that zygote also contained 46 chromosomes. This is it's kind of like, it's a pie graph that shows you the stages of the cell cycle. Interphase is in blue, and interphase is divided into G1, and G2. During G1, the organelles are duplicated because in addition to each new cell having to have a copy of the DNA, each new cell also has to have, if there's one mitochondria in the parent cell or the original cell, then there's got to be one mitochondria in the daughter cells that form. So the organelles have to copy themselves or duplicate. In the S stage of interphase, DNA replicates, okay? And that means DNA makes a copy. So the 46 chromosomes replicate so that each new cell will have a copy of the DNA that the original cell had. And stage G2 of interphase is when proteins are synthesized that are needed for cell division. You may remember what they're called. They're called microtubules or spindle fibers. Then we have the stages of mitosis, which are prophase, metaphase, anaphase, and telophase. And then we have cytokinesis, which occurs really kind of alongside telophase. So those are the stages that are the cell is actually dividing. All right, so for mitosis, make sure that you know it's defined as nuclear division in somatic or body cells. So all of our cells besides our sex cells that form egg and sperm, all of those cells, when they divide, when the nucleus divides and the chromosomes divide so that each new cell has a copy of the chromosomes, that division is called mitosis. Some cells are programmed to self-destruct, so the opposite of mitosis would be called apoptosis. That is programmed or genetically controlled cell death. Cells that are cancerous, cells that contain a virus, cells that just aren't supposed to be there as we develop. For example, we have webbing between our fingers and toes early on in development during embryological development. And that webbing, the cells that form that webbing, they're skin cells, and they undergo apoptosis so that we don't have webbed fingers and toes. That's one example. Meiosis is also cell division, but it is the process that results in sperm and egg or ova. So meiosis only occurs in the production of sperm and egg. All right, interphase is the period of time between cell divisions, and it lists what happens during G1S and G2, which I just went over with you. This shows you DNA replication, how the original strand of DNA unwinds and unzips, and then you see new DNA strands being copied from the original strand. So each new DNA strand is half old and half new DNA. All right, moving on though to mitosis. Mitosis occurs in 4 stages: prophase, metaphase, anaphase, telophase, and then cytokinesis follows. During prophase, now if you'll learn what happens during prophase, the rest of the phases of mitosis are pretty simple. But during prophase, you have chromosomes that look like X's. They look like the letter X because they're 2 rod-shaped chromosomes that are attached at a centromere, and the 2 strands that are attached are called chromatids or sister chromatids. So that's why they kind of look like the letter X. So the sister chromatids are the 2 identical copies of a chromosome, and so if they split apart, they're going to be identical in each of those chromatids will be identical in the new cells that form. Okay, so the nucleoli, remember those are the structures inside the nucleus that make ribosomes. The nucleoli disappear, centrioles move to opposite ends or opposite poles of the cell, and they start organizing their spindle fibers. The nuclear envelope disappears, and then the spindle fibers attach to the sister chromatids. Metaphase is really easy. The sister chromatids line up at the metaphase plate or the middle of the cell. Anaphase is when the sister chromatids are pulled apart. It says here daughter chromosomes, but before they're pulled apart, they're called sister chromatids. And then after they're pulled apart, they're called daughter chromosomes. But they're pulled apart, move toward opposite ends of the cell. And then in telophase, if you go back to prophase and you reverse everything, that's telophase. So the nucleoli that disappeared in prophase, now they reappear. The DNA that condensed into chromosomes, let's find where it says that. Chromosomes become visible, or they condense. In other words, we see the chromosomes instead of them being all spread out in the form called chromatin. So there are chromosomes in prophase, which look like the letter X, and then they uncoil and form back into chromatin, which is very, very spread out. You can't make out those X shapes anymore. So DNA uncoils in telophase, it coils up and forms chromosomes in prophase. And new nuclear envelopes or nuclear membranes form in telophase. And cytokinesis is the division of the cytoplasm, and it normally occurs during telophase. All right, and here are the stages. Here you've got interphase, where you've got DNAs in the form called chromatin, and so it's very, very spread out, not coiled up so that you can see the X shapes. Then you have prophase, when the DNA begins to condense into chromosomes. These centrioles move toward opposite ends of the cell. They form their spindle fibers. The nuclear envelope breaks down. The nucleolus disappears. Metaphase, the chromosomes that have condensed in prophase, they now line up at the metaphase plate. In anaphase, the sister chromatids or daughter chromosomes, whichever textbook you're reading at the time, but they're pulled apart. They were attached in metaphase. In anaphase, they're pulled apart by the spindle fibers. They're pulled toward opposite ends of the cell. And then you have telophase and cytokinesis, which kind of occur together. And you end up with two cells that are genetically identical to the parent cell. So this original cell is called the parent, and then the two cells that form are called the daughter cells. All right, last checkpoint question down here, number 28. What would happen if spindle fibers failed to form in a cell during mitosis? Well, the chromosomes wouldn't separate. The sister chromatids wouldn't separate because those spindle fibers attach to the sister chromatids and they pull them apart. So they wouldn't separate at all. Maybe one of the daughter cells would get all of the chromosomes and the other one would get none. I don't know, but they wouldn't separate. You wouldn't end up with two daughter cells that had equal numbers of DNA, equal amounts of DNA. They wouldn't be identical to the parent because those sister chromatids wouldn't have separated. Okay, little section on cancer and how that's related to mitosis. A tumor, which is also known as a neoplasm, is a mass produced by abnormal cell growth and division. If the tumor is benign, then it's not going to spread. And it's not life-threatening. It can be, but usually not. But it can get large. And if the tumor gets large, then sometimes it has to be removed. Malignant tumors are what we call cancerous. When someone is told that they have cancer, that means they have a tumor that is malignant, and it either already has spread or is capable of spreading from the primary tumor to other areas of the body. That's called metastasis. All right, an illness characterized by mutations that disrupt normal control processes and produce potentially malignant cells is termed cancer. And defined metastasis is when the cells that form a tumor spread to other areas of the body. And then lastly, we have this definition. All of our cells are not alike. They specialize to do different jobs, and we call this process differentiation. And this occurs when specific genes are turned off. So all the genes in our 46 chromosomes are not turned on. All of our cells have a copy of those genes, but most of those genes are turned off. The only genes that are turned on are the ones that are needed by that particular cell.